We are here today at Select Breeder Services in Chesapeake City, and we are going to start the first in the KWPN North America series of frozen semen for mare owners. Meet the founder and owner, Paul Loomis.
Welcome, Natalie, and I look forward to showing you our lab and what we do here at SBS. Our routine analysis post-thaw quality for all frozen semen that's frozen here, and any imported semen that we evaluate goes through the same process. What's important here is that it is done objectively. It is done blind by the person who's reading the motility on the CASA system. We do a post-thaw culture. We do a 30-minute post-thaw incubation, and we do a count on every sample. So Carly is thawing test straws right now from this ejaculate. So we empty the contents of the straw into a sterile tube, and from that we use a graduated inoculating loop to plate out a specific amount of the extended semen onto a blood agar culture plate, and those culture plates are then incubated for 48 hours, and we are looking for to make sure that there's no bacterial pathogens present in the thawed semen.
And you do this process on every stallion you collect here for freezing, but also affiliate labs.
Correct. Yeah. So everything we do here for freezing, every single ejaculate is evaluated this way. Every batch of semen that we freeze. The affiliate labs that are in the SPS network send us straws for us to do quality control on the work that they do. So we're also evaluating bacteriology, sperm concentration, and motility on the semen that they freeze. So any semen that's frozen by an SPS affiliate network lab undergoes the same exact process.
And you have those labs in the U.S., but also in Europe as well.
We have a few in Europe, we have 2 in Australia, and we have quite a few in North America.
Okay. And do you also run the same sort of these processes on semen you did not freeze?
We do. We have people that send us doses to evaluate as a third-party independent evaluation. We also do this process on any semen that we import for clients that want to know what the quality of that semen is when it's imported. What Carly's doing now is she's going to measure the sperm concentration. This is using an instrument called a nucleocounter. It's a fairly novel technique for measuring sperm concentration. It allows us to accurately measure sperm concentration in extended thawed semen by using a fluorescent dye, a fluorescent nuclear stain. So Carly has already taken and diluted the semen sample into a diluent that has a detergent in it. That detergent permeabilizes or punches holes in the plasma membrane of all the cells. The cells are then loaded into this cassette through these channels, and these channels are filled with a fluorescent nuclear stain called propydium iodide. The stain cells, the propydium iodide only goes into sperm with permeabilized membranes and only stains the DNA in the nucleus of the sperm head. So now this instrument has an ultraviolet light source, which is pointed at this viewing window inside the machine with a photodetector cell on the other side. And the only thing that this instrument sees is fluorescing sperm heads. So most instruments that measure sperm concentrations using a photometric technique are measuring how much light is blocked by the semen sample. And you therefore can't use those instruments with extended semen because the extender itself will block the light. So what this does is allows us very accurate counts of sperm cell concentration in extended semen.
And why is concentration so important?
We want to know exactly how many total sperm are in the insemination dose, because we're looking to calculate how many motile sperm are in the insemination dose, and so therefore we need to know the total plus the motility as well. In this particular sample, the motility, the concentration is 235 million per milliliter. Now, when we do post-thaw evaluations, one critical thing in our lab, we do them blind. And what I mean by that is one technician, in this case Carly, is selecting which samples she's going to thaw and make the dilutions in an extender. The person, in this case will be me, who's going to read the motility on the CASA system. I have no idea which samples she selected. We also we tend to do them in batches of at least 5 at a time. I'm going to thaw 5 samples. I have no idea what they are, what the quality is going to be, and then when they hit the 30-minute incubation time, I'll do an analysis of the motility on the CASA system. The other thing that we do as a control in our lab is we have what we call reverse QC or bioassay straws. So we freeze a large batch of straws, maybe 100 to 200 straws from one collection, very carefully. And then we will blindly analyze individual straws from that batch, at least 8, maybe up to 12. We can then calculate what the expected variability is between individual straws within a batch, because there will be some variability between individual straws. And there's also some variability between one analysis and the next analysis. That variability shouldn't be too much, and by analyzing enough individual samples, enough individual straws, we can calculate what the variability is, calculate the coefficient of variability for that batch, and then use those as controls. So whenever we're doing post-thaws like this, the person, the technician who's thawing the samples will select one of these straws and slide it in there somewhere. We may do 20 post-thaws every morning, and they may slide one in there. I have no idea, or the technician reading the CASA has no idea which one it is. But what it does is it provides for us a control to know that if that test straw doesn't fall within the normal range of what we expect for that batch, then perhaps there was an issue with the extender we diluted the semen in, an issue with temperature of the water bath or temperature of the stage, something systematic in our process that could affect the motility results for all the other samples that we look at that day. She's now diluted that sample into some milk-based extender, and then we're going to incubate the sample for 30 minutes in a water bath. So what we have found is that there are stallions who, if you look at the motility directly after thawing, it might be okay, and then by 30 minutes of incubation at 37, the motility falls off drastically. Most normal stallions with good semen quality and good fertility, the motility should be maintained at least out to that 30-minute time point. So when we report progressive motility in a thawed sample from our analysis, what it means is that's the progressive motility after dilution and a 30-minute incubation at 37 degrees. So it is a bit of a stress test, a heat stress test on the cells post-thaw that gives us a little bit better differentiation as to the quality of that sample.